Hello everyone, welcome to the course Experimental Biochemistry, which is coordinated by Professor Dasgupta and Professor Dev from IIT Kharagpur. I am Beas Mukherjee, a research scholar from Dr. Shomodev's lab, and today I will be taking a revision session for you. So in this session, we will go through all the topics that we have covered previously, and then we will be solving the MCQs and MSQ questions. Okay, so in today's class, I did not include the theory part because all of that has been covered until now. Questions are basically based on that. Okay, so the questions are of two different levels. There is moderate level question and there is your advanced level questions. We will begin the class 10 minutes from now. So let's get ready for the class now. Okay, so welcome back. Let us begin with today's discussion. So, in today's talk, we will be going through questions. Okay, MSQs and MCQs, and we will. This is just before your exam. So, all the best for your exam. And these kind of questions you can expect in your exam. There can be different kinds of questions, but my recommendation would be go through all the assignment questions properly, and these revision questions should also be helpful for you. Okay, let us begin with today's lecture. Okay. So in today's talk, we will be covering the topics pH, molarity and buffer concept, pKa, isoelectric point of amino acids, basics of proteins and nucleic acids, enzyme kinetics, chromatography and its types, and biophysical techniques, then purification of proteins and nucleic acids, and recombinant DNA technology. So these are the most important topics for your exams and also for your uh, competitive exams. So we'll thus practice these properly. Okay. So these are the moderate level revision questions. Okay. So the first question is, okay. So see, what is the molarity of a solution formed by mixing 100 ml of 0.5 molar HCl with 200 ml of 1 molar NaOH. So what would be the molarity of the solution when you are mixing half of acid and half of base? So let's see the answer. So for this, how do you need to solve it? See, the number of moles you're supposed to first calculate for both HCl and NaOH. So number of moles for H plus and Cl minus ions is 0.05. The number of moles for NaOH is 0.20. Okay, so which is more? If you see, the amount of NaOH is exactly double that of HCl, and therefore this excess number of moles, that is 0.15, this is the excess moles that we are having. So total volume of this final solution is going to be 300 ml, right? So therefore molarity is total number of moles divided by total volume, that is 0.15 moles by 0.3 liter, and that gives you 0.50 molar. So the correct answer is going to be B, 0.50 molar. For the next question, you have two questions here. So one question is, which amino acid would have the highest isoelectric point? The options are glutamic acid, histidine, lysine and aspartic acid. So which one is going to have your highest pi? And for pi, if you remember, you need to find out the pi using the formula pKa 1 plus pKa 2 divided by 2. Okay, and you should concentrate on the two pKa's that are just next to the pi, the isoelectric point at which the net charge is going to be 0. So you need to take those two pKa into consideration for pi calculation. Okay, for the next question, we have, in a denaturation experiment, the protein's secondary and tertiary structures are disrupted while its primary structure remains intact. So which of the following treatments is most likely responsible for this denaturation process? So your options are exposure to extreme pH condition. That is not the case. Heating the protein solution to 100 degrees centigrade. This is one of the reasons, but which is the major reason? So if this was an MSQ, you could have ticked this as correct. Okay. Adding a reducing agent as beta mercapto to ethanol. What is the fourth one? Treatment with a chaotic agent like urea or guanidine hydrochloride. So let us see. How can you disrupt the secondary and tertiary structures while the primary structure is still intact? And that is in a denaturing experiment. Remember, they have not mentioned SDS page or any other specific experiment name. Okay, so how do you solve this? For this question, you need to calculate the PI. This we have done many times. Okay, so you can get the examples in the previous lectures, and from there you need to calculate the PI for lysine, for glutamic acid, histidine and aspartic acid, and you will see that the one for lysine is the highest. Okay, so next is, in a denaturation experiment, the protein's secondary and tertiary structures are disrupted by. Okay, so the option number D is correct. Cheotropic agents disrupt the protein structure by interfering with hydrophobic interactions and hydrogen bonds that stabilize the folded conformation. This disruption leads to denaturation of the protein's secondary and tertiary structure, leaving its primary structure intact. Okay, so which is the next question? So that brings us to the next question, which is, which of the following amino acids is most likely to be found in the hydrophobic core of a protein? So which one do you think is found in the hydrophobic core of a protein? Well, tryptophan is the right answer. Tryptophan is a large hydrophobic amino acid with a bulky indoor ring. It is commonly found buried within the hydrophobic core of the proteins, where it contributes to stabilizing the protein's tertiary structure through hydrophobic interactions. Okay, so what are the other options that we have? Serine, this is not necessarily in the hydrophobic core. Lysine, more probability of lysine is to be on the surface because this interacts with water molecule, interacts with other proteins and ligands. Okay, what about glutamine? Same, it is a charged amino acid, so it has more probability to be on the surface rather than in the hydrophobic core. Okay, so in the next question, it is about recombinant DNA technology. So in this question, they are asking you, plasmids are often used as vector. Which of the following is not a characteristic of a plasmid? So circular DNA, we know that plasmids are circular DNA. Self-replicating, plasmids definitely have an ORI site. So this cannot be the right answer. Right, so it is not the right answer. Then presence of introns. Did you ever hear about introns in a plasmid? And finally, it is antibiotic resistance genes. Yes, this is a selectable marker which definitely we find in plasmids. What about the next one? The process of introducing recombinant DNA into the host cells is called translation, transcription, transformation and transduction. So the only option possible here is transformation. Okay, so one second. Yes, so we'll carry on with the questions. See, this is another question on your biophysical techniques. Okay, so a biochemist is investigating the kinetics of an enzyme catalyzed reaction. So which of the following techniques would be most suitable for measuring the initial rate of the reaction under varying substrate concentration? So which of these do you think is correct? A is gel electrophoresis, B is surface plasmon resonance, C is ITC, and D is spectrophotometric assay coupled with enzyme kinetics analysis. So the right answer is the last one, that is D, spectrophotometric analysis coupled with enzyme kinetics. Analysis is widely used for the study of enzyme kinetics. By measuring changes in absorbance over time, the initial rate of the enzyme catalyzed reaction can be determined, and under varying concentrations it can be calculated, allowing for the determination of kinetic parameters such as Michaelis-Menten constant and the maximum velocity of the reaction. Okay, so now that brings us to the next two questions, which are about cloning, right? Recombinant DNA technology. So there is Sanger sequencing and there is selectable markers in RDT. So which of the following steps is not the case in Sanger sequencing? So Sanger sequencing, if you remember, it happens by what? DNA replication, right? And the new strand gets your dideoxy nucleotides, and there the entire process of replication stops. Therefore, this is a process involving annealing of primers. Yes, definitely that is a very important part of replication. Otherwise, your synthesis of the second strand cannot begin. Then there is DNA polymerization, denaturation of DNA, and amplification of DNA. Denaturation, definitely, and DNA polymerization means addition of the DNTP onto the new strands to form the new strand complementary to the initial strand. So therefore, your amplification of DNA would not be the right answer. This is seen in case of PCR. Then what is the purpose of using selectable markers in RDT to identify recombinant DNA molecules? To visualize DNA fragments, to screen for the transformed cells, to quantify DNA amplification. So if you remember, when we were talking about transformation and recombination, we had very clearly distinguished between the recombinants and the transformants. So who are the recombinants? They are bacteria carrying your plasmid of interest, which is carrying your specific gene of interest. What about a transformant? Transformant is having bacteria with your plasmid of interest. So that means the plasmid has got into it, but it is not necessary that the gene of interest is present in the plasmid. So that is the difference between recombinant and transformant. Okay, and this selectable marker is present in the plasmid. It gets expressed and it gives resistance to antibiotics. So that means if the plasmid is present, the bacteria is going to survive in a media containing that antibiotic, otherwise it's going to die. Okay, it does not tell you any details about the gene of interest. So that means this is done to screen the transformed cells, but it has got nothing to do with recombination. Fine. So the recombinants, you cannot understand whether recombination has happened or not unless you go for sequencing. So now we move on to the next question. Okay, look at this explanation. So what do we see here? Explanation: Sanger sequencing method involves DNA denaturation, primer annealing, DNA polymerization, and chain termination using dideoxynucleotides, not amplification. And again, selectable markers such as antibiotic resistance genes are used to identify and select cells that have successfully taken up expressed recombinant DNA. Okay, we move on to the next question. Yes. So what about this question? HPLC, high performance liquid chromatography, or HPLC, contains a detector used for detecting non-UV absorbing compounds. So this is again a question on your protein purification, overexpression and purification, HPLC and other chromatographic techniques. Right. So what kind of detector is present in HPLC that can detect the presence of non-UV absorbing compounds? Diffractive index detector, fluorescence detector, mass detector and electrochemical detector. So the right answer is mass detector. So mass spectrometry is commonly used as a detector in HPLC for compounds that do not absorb UV light, providing highly sensitive detection and identification based on mass to charge ratios. Now we move on to the advanced level questions. So we don't have many advanced level questions, but let us try to understand them. So what will be the change in pH when 0.02 moles of HCl are added to 1 liter of a buffer solution containing 0.1 molar ammonia and 0.1 molar ammonium chloride? So the pKa of NH4 plus is given as 9.25. So let us have a look. Okay, so the right answer, see, decreases by 1.47, decrease by 0.47, increase by 0.47 and increase by 1.47. So this is a question that you can get in your GATE examination as a NAT or what, numerical some question, right? So you can get these kind of questions in your GATE exam. Excuse me. So how do we solve this? Let's try to understand. So to find the change in pH, we are going to use Henderson-Hasselbalch equation. So pH is equal to pKa plus log of the conjugate base divided by acid, right? So initially concentration of A minus is equal to 0.1 molar. Why? Because ammonia has been added, then it is concentration is 0.1 molar of ammonium chloride, right? So NH4 plus concentration is also 0.1 molar, and the concentration of your acid or HCl. Okay, so here this is actually the concentration of Cl minus because your conjugate base is Cl minus, right? So the concentration is 0.1 molar of both conjugate base and the HCl acid. Okay, now what happens? The pH is coming to be 9.25 if you solve according to Henderson-Hasselbalch's equation. When this ammonia is getting added, if you remember La Chatelier's principle, when the reactants are more, the reaction equilibrium shifts towards the product side, right? So more of the product will now get formed. If the product side is heavier, that means there are more products added, then the reaction equilibrium shifts towards the reactant side and more of the reactants will actually remain. Okay, so the reaction is basically going to slow down. So now see, when 0.02 moles of HCl are added, it reacts with NH3 plus to form NH4 plus. And NH3 plus HCl gives your NH4 plus Cl minus. This change in concentration of NH3 and NH4 plus is equal to the moles of HCl added, which is 0.02 moles. NH3 is equal to 0.1 minus 0.02. What is this 0.02? This is basically the change in concentration of ammonia and ammonium, which is equal to the moles of HCl added, and that is 0.02 moles, right? So what happens? The concentration of ammonia gas, that is, you're getting 0.1 minus 0.02 and that gives you 0.08 molar. Now what is the concentration of NH4 plus? Okay, so the concentration of NH4 plus is again 0.1 plus 0.02, because if you see this reaction, NH3 plus HCl gives NH4 plus and Cl minus. NH4 plus is getting formed because of NH3 plus and HCl, right? And that is why it gets added, concentration gets added, and we get 0.12 molar NH4 plus. So what is the pH? pH is coming as, if you solve using Henderson-Hasselbalch's equation, okay, your pKa you had calculated, that is 9 by 2.5 divided by log of, here who is the your conjugate acid, tell me, NH4 plus, this is the conjugate base, right? So conjugate base is NH4 plus divided by 0.08, which is the NH3. So then what happens? We get 9.72. And the change in pH is 9.72 minus 9.25, that is 0.47. And so the right answer is, it increases by 0.47. So there is a rise in pH by 0.47. Okay. Now we will move on to the next question. These kind of questions are very important. So try to practice as many numerical problems as possible. It will clear your basics and it will help you in your competitive exams. Okay. So for the next question, again this is a very simple question. What is the approximate pI of a dipeptide composed of glycine whose pKa1 is 2.4 and pKa2 is 9.6, and alanine whose pKa1 is equal to 2.4 and pKa2 is 9.7. So basically we are having these two amino acids forming a dipeptide, right? So pK1 and pK2 of both glycine and alanine are given. You try to draw it first, make the glycine in front and alanine after it, and then you find out the N and C terminal, you find out all the groups that can be ionizable, right? So find out the ionizable group, and then from there you calculate the pI of the dipeptide. If you remember our previous lectures, we had solved a question like this. We had done this for a tripeptide, and so it should not be difficult for you. Okay. Right, so we are solving questions today. This is a revision class and you can try to solve this question. Try solving, then I'll take away the answer slide in that case. So these are the advanced level questions. We have already covered the moderate level questions until now. Okay, so these are the advanced level questions. You can try solving this one. We had tried to solve this in the class, so I'm sure this shouldn't be difficult for you. And this entire lecture is getting recorded, so even if you missed the initial portion of it, don't be disheartened, because you can find that on YouTube. Okay, the link will be shared with you. The entire presentation slides, everything will be shared with you. So you can try solving this question. You can give me the answer in the chat box. Otherwise, you can tell me, we can carry on with the questions. Okay, whatever you want. So the PI calculation questions are very important, because these kind of questions will be asked in your exams. It can be the PI of a single amino acid. It can be the PI of a dipeptide or tripeptide. And you should be able to solve them because we have discussed all of these questions in class. Okay, so let us check the answer. So the answer is here to find the PI of a dipeptide. We take the average of the pKa values closest to the point where it carries no net charge. This we know. So the PI of a dipeptide would be the average of the pKa values for the amino acids it contains. So for glycine and alanine, the pKa values are 2.4 and 9.6 respectively. So then you are getting the average pKa as what? Now 2.4 plus 9.6 divided by 2. So here if you see glycine and alanine, there are no ionizable groups, right? So there is no question of pKa 3. There are only two ionizable groups. One is at the N terminal, one is at the C terminal. So N terminal is your NH2 group of glycine and yes, NH2 group of glycine, so that is 9.6, and in the C terminal of the dipeptide you have the ionizable group of alanine, which is 2.4, it is the carboxyl group. So 2.4 and 9.6, you get the average, and that is your isoelectric point. So always for solving these kind of questions, please make sure that you draw the diagram first. As soon as you draw the diagram of the dipeptide, it will become very clear to you. Okay, let's move on to the next question. So what method is suitable for determining the secondary structure content of a protein? So first option is isothermal titration calorimetry. The second option is dynamic light scattering. C, right, circular dichroism spectroscopy. Fourth option is your mass spec, and fifth option is electrophoretic mobility shift assay. To find out the secondary structure of protein, very good, circular dichroism is the right answer. Okay, so now we have another question. Which technique is used to study the dynamics and conformational changes of a protein in real time? So the first option is your electron microscopy. Second option is nuclear magnetic resonance spectroscopy. Third option is fluorescence resonance energy transfer. Fourth is your circular dichroism spectroscopy, and the fifth option is isothermal titration calorimetry. So which one do you think will help you to understand the dynamics? Now electron microscopy will give you things about the structure, right? So how does it look? So that is going to tell you about the structure. Fluorescence anisotropy is going to tell you about its function, right? Whether it is interacting with something else. So that is going to emit fluorescence. It is going to tell you about the proximity with another protein or ligand or a like monomer or something. So then after that there is circular dichroism spectroscopy. This tells you about the secondary structure. What about ITC? ITC tells you about the kinetics, right? It tells you about the energy of binding, free energy with the ligand, free energy of binding with the ligand. So nuclear magnetic resonance would be the right technique to study the dynamics and conformational changes of a protein in real time, because you can see the peak shift in NMR. Okay, so these two are right answers. Good. So we will move on to the next question. So which technique is suitable for investigating the kinetics of protein folding or unfolding transitions? Protein folding and unfolding. Okay. Circular dichroism spectroscopy. And when you have ITC, differential scanning calorimetry, fluorescence resonance energy transfer, and X-ray crystallography. So here the kinetics of protein folding and unfolding is being studied. What is the next question? Next question is, which method is most suitable for determining the stoichiometry of a protein complex? So, analytical ultracentrifuge, NMR, size exclusion chromatography, isothermal titration calorimetry, and fluorescence resonance energy transfer. Which would be most suitable? So in this case, you are saying that it is analytical ultracentrifugation. Is this for the second question? Right. Yes, very good. Okay. We'll have a look at the answers. Okay. So most suitable for studying protein complexes, in my opinion, should be analytical ultracentrifugation. Okay, but I will have to check whether this is correct or not, and for which technique is suitable for investigating the kinetics of protein folding and unfolding reaction. This should be differential scanning calorimetry. You can find out how it works. Okay, so I think we did not cover DSC in the previous lectures. So you can go through it. You can find out how DSC works. This is a very interesting technique. So you can definitely go through it. You can also read about differential light scattering. Okay. Right. So which of the following is a potential ethical concern associated with recombinant DNA technology? A is loss of genetic diversity. B is increased risk of genetic diseases. C is patenting of genes. And D is environmental pollution. So which of these is a major concern to the environment and humanity as a whole? What is the second question? The second question is, which of the following factors does not affect the retention time of a compound chromatography? So if you remember the retention time, it is the amount of time for which the compound is still there in the chromatographic column and it is not eluting. So what affects that? Column length, yes, definitely. Mobile phase composition, definitely, because when there is more ions in the mobile phase or there is some other component, then the retention will be changed, right? Because it is going to, because this protein is going to interact with the mobile phase components. Then there is detector sensitivity and sample concentration. So of course sample concentration also affects retention time of a compound in chromatography. So detector sensitivity should be the right answer in this case. Okay, let's look at the first question. Which of the following is a potential ethical concern associated with recombinant DNA technology? So, your option number A is loss of genetic diversity. That is definitely not the case. And then there is increased risk of genetic diseases. So, we are doing RDT so that we can decrease the risk of genetic diseases. So, this too cannot be the right answer. Patenting of genes. Yes. So, let's say someone has created a new gene, because novel genes are getting created every single day. So, if there is a gene created and someone is patenting it, then that is definitely very, very concerning. And what about environmental pollution? Environmental pollution is associated with any discipline, right? So yes, explanation: the patenting of genes raises ethical concern related to the ownership and control of genetic information, potentially limiting access to important technologies and discoveries. Okay, so there was a case with HeLa cells, which is the cancer cell line, and you can read about this. There was the story of Henrietta Lacks, right? Yes. So there is a story. There was the case of Henrietta Lacks, and she was suffering with cancer. And then after this patient was dead, she was dead, and by that time the doctor had taken out this tissue, cancer tissue from her body, and then he was actually working on it. So Then the patient's family actually claimed that this is illegal, this is unethical. You cannot take away something from our uh, dear and near one like this and go for research. And so that was a huge bioethical problem. So you can read about it. Every single cell biology book will give you a small account of HeLa cells. So this is a very interesting case, and since then the entire your intellectual properties and bioethics, all of these became very serious. Okay, so what about the second question, explanation? So detector sensitivity does not directly affect the retention time of a compound. In chromatography, of course, retention time is primarily influenced by factors such as the column length, mobile phase composition, and sample concentration. So that is the last slide of today's talk, and I would recommend you to go through the other slides. There were many questions. You can go through them. You can ask me. So these are the different topics that I had covered in today's talk. So you can ask me the questions. If you have any questions, you can ask me. If I'm not able to answer today, I will answer to you on Tuesday. That is going to be our last class. So we are going to learn about blotting. In today's lecture, I did not include questions from blotting because I haven't taught you that yet. So we have questions from all the topics that I've taught before. Okay. Yeah, sure. You can see the slides. Actually, the slides, these will be shared to you anyway. So this is first your introduction slide, and then this is your revision questions level moderate. Okay, so this is the first question. What is the molarity of a solution in which you are mixing HCl and you're mixing NaOH? So how do you find that out? This is the way you find it out. You calculate the number of moles of HCl and number of moles of NaOH, and then you calculate the total volume, and then molarity is nothing but total number of moles by total volume. That is how you get the right answer. Okay. For the next two questions, you have these two. Which amino acid will have the highest isoelectric point? So the all the four amino acids are given, their pKa values are given. You are supposed to calculate the pi. Okay, I'm sure you can do this. We have practiced a lot of these. And then in the denaturation experiments, they are asking you protein secondary and tertiary structures have been disrupted by, but then the primary structure is kept intact. And so in an experiment, how do we achieve that? So one of the options would be your heat, but then the more appropriate answer in this case, because this is a denaturation experiment, this is not SDS page. They have not mentioned SDS page, and therefore a better answer would be treatment with urea or guanidine hydrochloride, right? Because this is a chaotropic agent which is going to disrupt all the secondary and tertiary interactions of the protein. For the next one, you see we have which amino acid is present in the core of the protein, and so the right answer is tryptophan, because you know that tryptophan is very good at forming these pi-pi interactions, pi-cation interactions, right? Hydrophobic interactions. What about the other amino acids? Serine, lysine, glutamine, all of these are either polar or charged amino acid, and so they have more propensity to be on the surface of the protein and interact with water molecules or any ligand molecules. For the next two questions, we have um, which is not a characteristic of the plasmids, right? So plasmids, we know they are definitely circular DNA, they are definitely self-replicating, they have this ori site, but then we have never heard of any plasmid having introns, right? So definitely presence of introns is not a required property to be a plasmid. To be a vector, right? And antibiotic resistance gene. This is a selectable marker and you definitely need it. So, what about the next question? The process of introducing recombinant DNA into the host cells is called. I am sure you know the answer. Yes, it is transformation. So these are the two answers, right? In the next one, there is a small experiment. Yes, right. So you're even answering. That is very good. No. The process of introducing recombinant DNA into host cells. It is transformation. Translation is synthesis of protein. Right? Okay. So let's look at the next experiment. So a biochemist is investigating the kinetics of an enzyme catalyzed reaction. Which of the following techniques would be most suitable for measuring the initial rate of the reaction under varying substrate concentration? So he is measuring enzyme catalyzed reaction. There is gel electrophoresis. Now varying substrate condition, right? So there is enzyme gel electrophoresis, but then what can you see in gel electrophoresis? Then there is surface plasmon resonance. Surface plasmon resonance tells you how tightly the enzyme is maybe binding with the substrate, but will it tell you about the changes in kinetics of the enzyme under varying substrate concentration? Then there is ITC, which is again going to tell you about the energy of binding. And finally, it is spectrophotometric analysis coupled with enzyme kinetic analysis. So to understand about an enzyme, you need to perform kinetic studies on the enzyme. Correct? Right. So see, spectrophotometric analysis could be coupled with enzyme kinetic analysis. So by measuring changes in the absorbance over time, the initial rate of the enzyme catalyzed reaction can be determined under varying substrate concentration, allowing for the determination of kinetic parameters such as Km and Vmax. Okay. So for the next question, we have which of the following is not a step in Sanger sequencing method? So in Sanger sequencing, what happens? One DNA strand is given and you synthesize another DNA strand, but whenever there is a DDNTP, synthesis stops. So it is actually replication but not amplification. Therefore, annealing of the primers is definitely very important, because without annealing, you cannot have extension and you cannot create a new DNA molecule. There is DNA polymerization. Polymerization means that the DNTP molecules come and bind to the DNA, right? And that polymerizes the DNA. Then we have denaturation of DNA, of course important, and amplification of DNA. This is not important, right, in Sanger sequencing. So what is the purpose of using selectable markers in recombinant DNA technology? If you remember, we had discussed about the difference between recombinants and transformants, right? So recombinant is a bacteria which contains your plasmid of interest plus your gene of interest in that plasmid. That is a very good. Yes, to screen for transform cells. Very good. So, but what about your transformants? Transformant is a bacteria which contains your plasmid of interest only. It may or may not contain the gene of interest. That is the story, right? And this antibiotic resistance gene is going to be present in the plasmid. So, if you have the plasmid, the bacteria survives in the growth media with antibiotic. If you don't have the plasmid, it dies. So, therefore, it is good for screening for the transformants, not recombinants. Okay. Next, this is the explanation. You can see they are talking about DDNTP and all the different steps. Fine. Now for the next question, we have HPLC. So in HPLC, if there is a substance which cannot absorb UV light, then how do we measure the amount of the substrate or whether it is present or not? Right. So for that, we need to go for what? Refractive index detector, fluorescence detector, mass spectrometer, or electrochemical detector. right answer should be C, mass spectrometer. Okay, have a look at this. So mass spectrometry is commonly used as a detector in HPLC for compounds that do not absorb UV light, providing highly sensitive detection and identification based on mass to charge ratios. Then we have the advanced level revision questions. So this is a small numerical in which they are asking you if HCl, so see if 0.02 moles of HCl is mixed with buffer, right, solution containing 0.1 moles of ammonia and 0.1 moles of ammonium chloride, then what is going to happen? So for this you need to remember the Le Chatelier's principle, and you know that when what happens here is that when you're adding something which is supposed to be on the product side, then what happens, the reaction equilibrium is pushed towards the reactants and it slows down, right? But when you're adding something that is supposed to be on the reactant side and the reactants are now more, number of moles of reactants are more than that of the products, then what happens, the reaction equilibrium shifts towards the product and the reaction fastens, correct? So that is what you need to keep in mind. You need to solve this using the Henderson-Hasselbalch equation. You can go through this and you can be solving this. Alright. So for the next question, we have, since here I think you were already there and you had already seen, right? So dipeptide, glycine and alanine. Thank you so much. All the best for your exams. If you have any questions, you can ask me today or the next day, that is Tuesday. Okay. Thank you so much. So I'll be ending the class now, if that's okay. Okay, great.
Mm.